Hello everyone, this is Dr. Nidhi from Biotechnica. In today's video, we'll discuss a very interesting topic named Lac Operon. Yes, Lac Operon, this is one topic which you will find in the syllabus of CSIR exam, GATE exam, as well as in the syllabi of other exams. So basically, this topic belongs to another topic which is gene regulation, right? So firstly, we must know that what do we mean by gene regulation. So let's get started from here only. Gene regulation means that the machinery, the system which works to regulate the level of gene expression. It will decide that whether a gene will get expressed, whether it will turn on or it will get repressed. That means it will be turned off. Okay, so based on that, whether the genes they are being regulated for the gene expression or not, we can keep them into two categories. So here, as you could see, the two categories which we have here are housekeeping genes and regulated genes. Okay, now what are housekeeping genes? So the genes which will be responsible for showing the constitutive expression. Constitutive expression means that they have to express all the time. Since they are involved in the production of the enzymes which might be responsible for some central metabolic processes happening in the cell. Like I give you a very simple example for that, respiration. The enzymes which are involved in the respiration, so they have to be available all the time in the cell. So the genes which will be responsible for producing the enzymes which are part of the respiration process, they have to constitutively express. So genes are transcribed continuously. Okay, so therein we can underline genes are transcribed continuously. Okay, now on the other hand, we may have some genes which could be regulated, means their expression that is being controlled. They will be getting expressed only when their products, the enzymes, they are needed. So that will depend, right, whether the cell is in need of those enzymes or not. So yes, what are the properties of the gene which are being regulated? Regulated expression. Genes are transcribed when the gene product is required. Right, now here if I give you an example, so DNA repair, right? Now the enzymes, the proteins, the factors which are part of the DNA repair mechanism, they have to be produced, they will be required in the cell when there is a need for any DNA repair mechanism to get started, right? So let me tell you that the regulation at the gene expression can happen at various levels, right? Starting from the transcription till the availability of the protein, the regulation machinery can work. However, first and principal stage in gene expression will be transcription. Make sense? Right? Transcription is the first stage where the gene is getting expressed. So if the gene that needs to be repressed, if the proteins, the enzymes which are coming out of the gene, they are not required, so transcription that should be stopped. So this is the reason that we'll be considering it as the first and the principal stage in gene expression. Now moving further, Operon. Okay, now let's come to Operon because we'll be learning Lac Operon. But before that, let us see that what actually we do mean by Operon, right? Operon, that is an example of gene regulation at the transcription level, right? So if we need to simply define Operon, so how we can do that? Operon, that is a cluster. It will be comprising of a cluster of genes which will be under the control of a single promoter. Means the expression of the genes, the structure genes which are part of the operon, that will be controlled under a single transcription event. Whenever they'll be transcribed simultaneously, because usually the enzymes which will be coming out of these structure genes, they will be common to a central metabolic pathway, right? Now, operons, basically, operons, these are the characteristic of bacteria, right? So, bacterial genes, they are often found in operons. This is how the operon would look, right? If I just zoom in to this image, so you can look here that there are 3 genes. So these are the structural genes. Now that will depend upon the type of the Lac Operon will have, type of the operon will have. Like in the Lac Operon, we have 3 structural genes. Okay, some other operon system that may have 5 or even more than that. So that will depend upon the nature of the operon system. So here now you can understand that whenever the genes, these 3 genes will be getting expressed, so that will be under the single transcription event. Now let us move to our Lac Operon, right? Let us talk about the Lac Operon. Now Lac Operon, that is an example of the inducible operon. So firstly, we must understand that what do we mean by inducible. Now, like I told you that gene regulation, now genes, they can be turned on or off. When it comes to regulation, they can, their level, their expression level, it can be enhanced or it can be repressed. Okay, now the operon system, which will be generally in off state, right? And they need to be turned on whenever some inducer molecule will induce them, right? So that kind of operon system is known as an inducible system. Lac Operon is the example of an inducible operon, right? Here, the genes which are part of the Lac Operon, 3 structural genes. So we'll be talking about shortly. Okay, we'll be talking about them shortly here under our discussion. So I'll tell you that what are those 3 genes and what are the products which will be coming out of them, how they are important in metabolizing lactose, right? So now we understand that the Lac Operon, it has to be induced, okay, and this Lac Operon is significant because the enzymes which will be produced, they are required in the metabolism of sugar lactose, right? Now lactose, that is a disaccharide, okay, and it has to be metabolized, it has to be processed into simple sugars. So whenever the bacteria will not have the option to utilize glucose, okay, and it has the availability of lactose only, so anyways it has to survive, it will be using lactose, and then lactose has to be processed, okay, but that will be there in only that condition when bacteria doesn't have glucose, okay, it has to use lactose. I hope you are getting the point. Means if it has to use lactose, it must contain the enzymes which are required for processing the lactose, right? Otherwise it will not be in need of those enzymes. So that makes sense. When those enzymes they are needed, Lac Operon will turn on. Okay, when those enzymes are not required, so the Lac Operon system will turn off, right? So here we'll talk about the Lac repressor. Lac repressor, that is responsible for keeping the Lac Operon in off state unless and until they are required for producing the Lac enzymes. So what will happen in the absence of lactose? When bacteria, they are not having the availability of lactose, and the system that has to remain in the off state, okay. So here, see, have a look over this diagram. So here we need to learn about the Lac repressor. See, Lac repressor is the protein. This is the image, just have a look over this. So Lac repressor, basically this is responsible for keeping the system off. Now, if we look at the structure of the Lac Operon, so these are the 3 genes, right? Lac Z, Lac Y, Lac A, and here you will find that we do have the regulatory gene. Okay, now this regulatory gene is responsible for producing the Lac repressor. Lac repressor, that is the protein which is responsible for keeping the Lac Operon system off. Okay, until and unless there is a need of the enzyme. So this repressor protein, basically it binds at the operator region. So this is the operator region. Okay, so if we just need to know about the Lac Operon, so if I just tell you about the basic structure, so 3 structural genes we do have here, Lac Z, Y, A. Apart from that, you will see this is the operator region where Lac repressor binds, and this is the promoter region. Okay, for any transcription event, there will be a promoter site where the RNA polymerase will bind to start with the transcription. right? Now, what is happening when the lactose is not available? This lac repressor, which is being produced from the regulatory gene. Now, this regulatory gene is also part of the Lac Operon. So, we do have the structural genes, we have operator, we have promoter, and then this regulatory gene, which is producing this repressor. Now, when there is no lactose around, we can see this repressor will be binding at operator site. Yes, please note it down that the repressor protein binds to the operator, not at the promoter. Okay, it will bind to the operator, however, it will block the promoter for being available to RNA polymerase, and hence the transcription that will be ceased. This is when there is no availability of lactose. Okay, even before that, let me show you that what is the significance of these three structural genes, right, which we have seen in the structure of the Lac Operon, which their products, they are required to acquire and process the disaccharide lactose from the environment. So like I had mentioned earlier, that when you will be having the bacteria will be having the lactose around, only in that case the system will turn on, because that those enzymes, they will be required to break down the lactose into simple sugar. So like we have seen in the diagram that there were three structural genes. So LAC-Z, that encodes beta-galactosidase. So this is the main enzyme, right, which will be produced from the LAC genes, which is responsible for breaking lactose into simple sugars. Then we have LAC-Y gene. LAC-Y gene, that is responsible for producing beta-galactoside permease. Now, if you ask that what is the role of beta-galactoside permease, so let me tell you that this enzyme, this is the membrane protein, okay, and this is responsible for transporting the lactose into the cell, right? So what we have learned that the LAC enzyme, they are responsible for acquiring and processing the lactose. So basically this is about acquiring the lactose, right? So lactose has to get entry into the cell, and then only it will be processed. So here you can see this is the permease enzyme, which will be responsible for allowing the lactose to enter. And next you will see that beta-galactosidase, that is responsible for processing the lactose into galactose and glucose. Apart from that, let me tell you that beta-galactosidase is also responsible for converting lactose into alternate form of it, which is known as allolactose. So precisely this is allolactose and not lactose, which is the inducer of Lac Operon, right? So in allolactose and lactose, now what is the difference between allolactose and lactose is that lactose, that is made up of glucose and galactose. In allolactose, you will find that there is beta 1,4 glycosidic linkage between glucose and galactose, whereas in lactose you will find this is beta 1,6. So that is the difference between these two. Now, moving further, let's talk about LAC-A, right? So LAC-A encodes beta-galactoside transacetylase, right? Now, this enzyme, beta-galactoside transacetylase, this is of no biological significance to the Lac Operon. So as such, it is not involved directly in the processing of lactose. So now we know that what all three enzymes, they are produced when the Lac Operon system will turn on. Now let's move further. So this is where you can see lactose and allolactose, they have been shown. This is lactose, this is allolactose. So lactose that is converted to allolactose, that also happens with the help of beta-galactosidase enzyme. All right. Now moving further, let's talk about what will happen when there will be availability of lactose. Okay, so far we have learned that when there will be no lactose around, how the repressor will keep the lac genes in off state, right? So I hope you got an idea. We have a repressor which is coming out of the regulatory gene, will bind to the operator, will not allow RNA polymerase to bind to the promoter, and hence the transcription will not happen. However, when the lactose will be available, okay, when lactose is available, let's see now how the system will turn on, right? Here, when this lactose is available, and as we have learned that lactose will be converted to allolactose with the help of beta-galactosidase. Now, you may ask that beta-galactosidase is not there because system has not been turned on, right? So, where this beta-galactosidase is coming from? So, let me tell you that very little amount of the enzyme that will always be present in the cell. So, that amount of enzyme will be sufficient to convert this lactose into allolactose to initiate the Lac Operon. Okay, so lactose will get converted to allolactose, and this allolactose, as you can see, this uh, small circular shape which has been shown in this figure, this is indicating what? Allolactose molecule. So this allolactose, it is binding to the repressor. As soon as this repressor will bind with the allolactose, it will undergo structural changes and it will not be able to bind to the operator site, which it was doing earlier. Okay, so when it will not be binding to the operator site, that means operator will be all free, promoter will be all free, and hence RNA polymerase will be able to bind to the promoter and the transcription will happen. Now you can see that transcription is going on and all the three enzymes, they are being produced. So in this way, the amount of beta-galactosidase will increase in the cell. More and more lactose will get converted to allolactose. Not only that, the lactose that will be simplified, right? That is the main reason for which these genes, they have been turned on, right? So I hope now you got an idea that what all will be happening when there will be no lactose around and when there will be availability of lactose for the bacteria. So the system will turn on because allolactose, which is coming out from the lactose only, this will be turning the system on by acting as the inducer. Now we are left with another part of Lac Operon, which is equally important to be known, and that is about the cap protein, right? Cap protein is an activator. So far we have talked about the repressor, right? How repressor was having an effect over the expression of the Lac gene. Now let's talk about the cap protein. So cap protein, which means if we just expand cap, so what does it mean? So catabolite activator protein, right? It acts as the glucose sensor. Now, we have learned that what would happen when there will be availability of lactose and when lactose will not be available. But how the system will respond when or the bacteria, in fact, will be having the availability of glucose. Okay, even if I ask you that what will happen, how the system will act if the bacteria has both glucose and lactose to be used, how it will decide that whether it has to use glucose or lactose. So let me tell you that bacteria will always prefer using glucose over lactose because glucose is the simplest sugar, and definitely bacteria will not be turning on the system the Lac genes if glucose is available to be used as the carbon source. Okay, now this catabolite activator protein, this is responsible for sensing the level of glucose, right? This is an activator. Since cap protein is an activator, it is known to activate the transcription of the operon system, and it will be turning on the system. It will help the system to perform only when glucose levels are low. Yes, so please note it down, right? This cap protein will sense the level of the glucose and it will help the Lac Operon to perform only if the glucose levels are low. That means till the glucose is present in ample amount, till the glucose is available for the bacteria to be used, cap protein will not turn active. Okay. So there are two things. One, we have learned about the repressor. Now let us learn that how the activator is influencing the Lac genes. Here, moving further, so like we have discussed, cap protein will be sensing the glucose level. So it senses glucose indirectly through the hunger signal. Hunger signal. Now, what do we mean by hunger signal? CMP molecule. Yes, cycloadenosine monophosphate, they'll be acting as the hunger signal. So basically what happens when there is a high amount of glucose available, right? So this glucose, it inactivates the enzyme adenylate cyclase, and hence ATP that doesn't get converted to CAMP, right? And the CAMP, which act as the hunger signal, they will be available only when glucose level that goes down, right? Means when the glucose is low, the adenylate cyclase, since it will not be getting inactivated, so it will convert ATP to CAMP. Now these CAMP, they will be binding to the cap protein, okay, and then this assembly of CAMP and cap, that will turn active, and it will be binding to the cap binding site over the Lac Operon, and it will act as the activator, right? So what we have learned, that the amount of glucose, when glucose is high, remember CAMP, that will remain low, okay, and if glucose comes down, so CAMP level, that will be increased, and they'll bind to the cap protein, they will turn it active. What next? So this is how the Lac Operon will look like, right? So we have already learned that there are 3 structural genes, then we have operator, promoter, and operator, as we could see that operator is overlapping with the promoter. So whenever the repressor will be binding to the operator, so that is how it blocks the promoter from binding to RNA polymerase. Okay, and now near to it, you can see we have the cap site, where this active cap protein will bind, but that will happen only when glucose is low. So now there are 2 things which you should know, that when glucose is low, cap protein, that will turn active and it will bind at the cap site. Okay, and if lactose will be available, so we'll have allolactose, which will be removing away the repressor, and hence the operator site will be all free and RNA polymerase will bind here. Okay, so these 2 conditions, they should be fulfilled if Lac Operon has to work. There are 2 ideal conditions, that lactose should be available and glucose, that should be either it should be absent or it should be there in very low concentration. Okay, so in this way, these events, they will be supporting the transcription event to happen, and these 3 structure genes, they will get transcribed. Okay, so just have a look over this figure. Here you can see this is the active assembly of cap protein binding to the cap site. Okay, and glucose, that is known to inhibit the adenylate cyclase, this enzyme, and hence the formation of CAMP, but only when glucose goes down, so we find CAMP to be available, which will be turning cap protein active, right? So 2 aspects of the Lac Operon we have learned, and I have told you that what are the 2 ideal conditions which are required for the Lac Operon system to turn active. First, there should be availability of lactose so that the repressor could get removed, and second, that glucose, that should be absent or it should be present in very low amount. Okay, now let us practice one question. If you have understood that what are the conditions which will be favoring the transcription of Lac genes, so have a look over this question. The Lac Operon in E. coli is controlled by both the Lac repressor and the catabolite activation protein. In an in vivo experiment with Lac Operon, the following observations are made. So have a look over 3 observations which have been provided in the question. CAMP levels are high, repressor is bound with allolactose, cap is interacting with RNA polymerase. So which of the following conclusion is most appropriate based on the above observations? So let's see that what are the 3 observations. CAMP levels are high. Yes, CAMP levels are high. That means glucose, that should be absent. Okay. Repressor is bound with allolactose. That means lactose is available. It is present. Cap is interacting with the RNA polymerase. So again, it indicates that glucose is absent. Only in that condition, we may have enough amount of CAMP available, which will be binding to the cap protein, turning it active, and cap will bind to the cap binding site, and it will support the RNA polymerase for transcription. So looking at all these 3 observations, we can easily make out that they are favorable for the transcription to happen from the Lac genes. So what should be our right option here? glucose is absent and lactose is present. That is option 4. I hope that was an easy one. So I hope you got an idea that how Lac Operon system works. So I hope that you will like the video, and please don't forget to post your comments if you would like us to come with more informational video like this. Thank you for watching the video.